you, Prisha, and welcome International Fellow based at Mahidol Oxford Tropical Medicine Research Unit in Bangkok, Thailand. It is an honor to be shortlisted for Nature Awards for Inspiring Women in Science and also be inspired by many wonderful colleagues. My research focused on a neglected tropical disease called melioidosis, which is caused by an environmental bacterium Burkholderia pseudomallei. Patients are often infected through direct contacts with contaminated soil and water, and once infected, half of cases died, and currently there's no licensed vaccines available. My teams use genetics to identify markers in both the bacteria and the host that are associated with greater disease risk. And one of our exciting findings is that the genetics markers in the bacteria associated with the disease-causing isolates has been selected as a vaccine candidate. We also collaborate extensively to develop a DNA-based rapid diagnostic test so that we can detect melioidosis patients faster, treat them sooner, and hopefully reduce the mortality of the disease. Thank you so much.
My name is Lana Josipovic. I'm an assistant professor at ETH Zurich, Department of Information Technology and Electrical Engineering. As our computational demands are constantly increasing, we need to invest in new ways to go about computing. Hardware accelerators, essentially digital circuits designed for a particular computing task, are very promising here as they can achieve high parallelism and good energy efficiency. Yet, designing, verifying, and using these custom accelerators are all very complex and time-consuming tasks that are restricted to a very limited number of hardware design experts. This prevents us from fully exploiting the computing capabilities of hardware acceleration. My research goal is to make the design of high-quality hardware accelerators broadly accessible, fast, and reliable. By exploring synergies across computer science and electrical engineering, I aim to revolutionize the way we go about hardware design and enable different users and different computing tasks to reap the benefits of hardware acceleration.
Hello, my name is Ana Benitez Mateos, and I am Ambizione Group Leader of the Department of Chemistry and Applied Biosciences in ETH Zurich. My research aims to develop more sustainable and efficient enzymatic methods for the synthesis of the molecules that we use in our daily lives, pharmaceuticals, food additives, and cosmetics. The problem is that enzymes are not adapted to the harsh conditions that might be required in industry, such as high temperature. To overcome this challenge, we apply a multidisciplinary approach, starting from bioinformatics and protein engineering to design more stable enzymes that will be then immobilized into customized materials for their final integration into the bioreactors. Our research will have also a broader impact in other fields where protein stability is a key factor, such as biomedicine, biosensing, or bioremediation. We work towards a more sustainable future based on a circular bioeconomy model.
Hello, I'm Kelly Nguyen. I'm a group leader at the MRC Laboratory of Molecular Biology in Cambridge. My lab studies this region of the genome called telomeres. Telomeres are located at the ends of chromosomes, and they're very important for protecting our genomic information. Their dysfunction has been linked to cancer and aging. What we aim to do is to understand how cells maintain the telomeres and how disease mutation give rise to telomere dysfunction. One of the very important pieces of equipment that we use for our research is a cryo-electron microscope. In the past few years, we have been using this machine to visualize molecular machines that are involved in telomere maintenance in great details. Our research has greatly advanced our molecular understanding of telomere maintenance and also give our insights into how disease mutation give rise to a number of diseases in humans.
Hi, my name is Simone Schöle and I'm a professor for medical microsystems at ETH Zurich in Switzerland. I'm an engineer by training with a PhD in microrobotics, and my team and I develop diagnostic and therapeutic systems at the micro and nanoscale to tackle a range of challenging problems in healthcare. To accomplish this, we leverage the convergence of several disciplines, such as engineering, physics, chemistry, material science, and biology. Just to give a brief example, on the therapeutic side, we aim to improve drug delivery. For example, in cancer therapy, only a tiny fraction of administered drugs actually reach tumor sites. So here we engineer biohybrid microrobots that incorporate live bacteria empowered with magnetic nanomaterials. They can carry drugs, trigger immune response, and can be enriched at targeted tumor sites via external magnetic control. We also design and engineer tailored magnetic control systems to allow precise targeting at human scale to reduce systemic side effects. I have worked on one of such systems during my PhD, which is today commercially available. So looking forward, I really aim to continue to push translational sciences with my team. Next to my passion in science, I love to hike and swim and run, but first and foremost, to play with my 2 little kids and spend quality time with my family.
Hi, I'm Caroline van der Sant, a biomedical researcher at the Doherty Institute and the University of Melbourne. For as long as I can remember, I've been intrigued by the fact that the same virus can result in mild infections in one person and have devastating outcomes in someone else. About 1 in 5 people worldwide suffer from impaired immunity, with aging being a major contributing factor. By 2050, around 25% of the world's population will be over the age of 65, increasing their risk for severe disease outcomes. It is therefore of the utmost importance that we find new and effective ways to protect them. Unfortunately, traditional vaccines and treatment strategies often focus on boosting a healthy immune system rather than restoring immune function, and therefore provide limited protection in high-risk populations. I lead a group of young and exceptionally talented researchers to tackle critical questions that will help us understand why some individuals fail to generate protective immunity, while others perform better than expected.